let's come back to our friend here, the agarose gel, to discuss some issues about cropping. So there's going to be scenarios in science when you decide you ran the experiment. Let's say each one of these was a different thing. Imagine you ran an experiment and you had item 1, item 2, item 3, item 4, and you loaded these separate items into these lanes. And you run the experiment and you decide at some point lane 3 is not pertinent. So you decide, I want to get rid of this lane 3 because I think the most relevant comparison is lane 2 to lane 4, and lane 3 is not contributing, and it's actually inhibiting my ability to compare 2 to 4. Now, this is a scenario that's going to come up a lot as a scientist, and you have the capability to do things like this. It's actually your responsibility as the scientist to determine what are the most relevant comparisons, okay? And then it's the reviewer's responsibility when you submit this paper. It's the reviewer's responsibility to essentially determine whether you have made that determination correctly. So I just want to say clearly that this is within your rights to do this as a scientist. It's within your rights to say, lane 3, that's not even relevant to what I'm trying to discuss here. I really need to compare lanes 2 to 4. And so you want to recreate an image that makes it better to compare 2 to 4. Now, you can do this with the cropping, but you have to be extremely careful here, okay? And one of the things that you need to point out is you need to make it clear that you have made this modification, okay? Because now we're going to be changing actually like the data within the box, okay? So you might want to do something like this. You want to get rid of that lane 3, okay? So what you might do is you might go to that data layer. You might select it, and you might erase. Okay, we haven't broken any rules yet. Now let me show you how to do this properly, which you can do, okay? Now what you can do is you can take the rest of that image, you can slide it over proportionately. Oh, it just got messed up. Zoom in. Now here's where rules cannot be broken. You must leave a space here between. Let me just relabel the lanes accurately quick here. This is now lane 4. Oh, because lane 3 was cut out. Now we can do the comparison. We can compare 2 to 4. And what you would write is in the legend or of the figure or underneath in the methods, you might write this particular gel was cropped, but it's derived from the same gel, and therefore comparing lane 2 to 4 is legitimate. Okay? And we make this known to the reviewer by leaving a clear distinction, this white bar, between the area where we cropped out. Okay? We're not hiding anything. The reviewer knows we've cropped something out, and the reviewer can ask what was cropped out. And in many, many high-impact journals, this is required. In many high-impact journals, it's required that you then turn in the image as well of the original uncropped image so that the reviewers can actually see what it was there that you cut out. But this is within your realm of potential. You can do this as long as you do it properly. Let me show you what you cannot do. What you cannot do is take that gel and move it over such that it looks like there was no crop there. This is illegal. Illegal. Let me write this in red. What's going on? Illegal. That is illegal. You cannot do that. You always must mark for the reviewer where a crop was made, if it was made. You can only crop like this. Understood? Good. Now let's talk a little bit more about cropping, okay? Because we might get into a scenario where a similar determination is made as a scientist. It might be that this band right here, this bottom band, this band might be the important band. It might be this band right here, this one, that is the pertinent data, okay? And this band might be something else. It might be a non-specific product. Who knows? It could be something that you determine is not relevant to the data. Now, again, this is a very, very nuanced issue in science, because as soon as you start to say things like this, you start to set yourself up where a reviewer might disagree with you. A reviewer might say, no, you're wrong. This is relevant to the data. This is extremely relevant to the data. And so if you crop that out, so you might envision a scenario where you decide you're going to do that. You're going to crop the image, select inverse, and you're going to crop the image. You decided that that data was not relevant. You're going to crop that out. And this is what you're going to present in your figure. This is a nuanced issue. You have to be extremely careful. And you will see this. You will see people do this. If you look at any Cell publication, you'll see all kinds of gels where they do this. Okay? They crop out things that are not pertinent. They crop out stuff, and you don't necessarily always know what they're cropping out. And you don't necessarily know if what they've cropped out is relevant or not. You don't know that as the reader or as the reviewer. So keep in mind that if you do this, you're setting yourself up to get in trouble. Okay? So, but there are situations where you can do this, and there are situations where this makes the data actually easier to interpret. So, like, what rules should you follow? What should you do? What should be your heuristics? Now, this is my heuristic. My personal heuristic about this is if there's data that is within the region, you probably want to include that data. So I never crop things real tight. I always get suspicious when people crop things really tight, if they were cropping like this, okay? I always sort of leave a little bit so that people can see what's on the top and the bottom, okay? So if you were going to crop this image, say, I would say you could crop like this. Select inverse. You could crop like this. Because these big bands, sometimes it's not always up to you to determine whether or not this is pertinent. It might be up to the reviewer. Okay, so you leave these things in so the reviewer doesn't get suspicious and doesn't think, if you're cropping like this, the reviewer's going to get suspicious and they're going to think, what are they not showing me? If you do this and you present it like this, the reviewer knows, okay, they're kind of showing me everything there is to see here. Okay, and you're going to sort of build a good rapport with the reviewer and with the editor of that journal. So I would suggest that a good heuristic is to always include as much as you can that you think might be pertinent. But it's within your realm of possibilities to crop out things that are not pertinent. And it needs to be you as a responsible scientist that makes that decision, but also reports the things that you've done in the methods or in the legend of these particular figures.